I'm Stuart Waugh, the New York State Center for Clean Water Technology. And for this month's monthly seminar, the center is really pleased to have Carrie McDonough from the Department of Civil Engineering here at Stony Brook University. Carrie has a very distinguished academic background, graduated in chemistry from MIT, subsequently went to the University of Rhode Island and got a degree in chemical oceanography, and then was a postdoc at Colorado School of Mines. She has joined the faculty and the Department of Civil Engineering at Stony Brook University, and we're very pleased to have her give the presentation for this month. Carrie, I'll turn it over to you.
Great, thank you so much for that introduction, Stuart. I'm very happy to be here today. Thank you for having me. So today I'm going to be talking about some of the work that I'm kind of building in my laboratory. I've been here since 2019, so we're kind of just getting started on a lot of exciting projects related primarily to PFAS mixtures and the ways that per and polyfluoroalkyl substances contaminating our environment can then lead to human exposure. So to get started, I'm just going to cover, a little bit of background on what these compounds are for those who might not be familiar. So per and polyfluoroalkyl substances is abbreviated Pfases, or some people use PFAS, same thing. These are extremely stable oleophobic compounds. They have many uses, particularly when you want things to be resistant, stain resistant, wax, grease repellent, waxy kind of materials, things like that. So lots of uses in industry and commercial products, Teflon tape, Teflon lined pans, things like that. Also used, a lot in foods that might be greasy to keep them from getting too saturated, things like microwave popcorn, as well as other kind of protective materials, Scotch Guard, those little kind of waxy papers they put around fast food when you get takeout. Carpets can be stain repellent, treated with PFASes, and also garments, so things like raincoats, rain boots, things that you want to be water resistant, have often been treated with PFASes. Additionally, there's a lot of uses in industry. One of the kind of most well-known and most widespread is the use of aqueous film-forming foams. So these are firefighting foams used to fight Class B fuel fires, and they've been used for a long time. They use them both in the training activities as well as to actually fight fires. So they've been stored and used throughout the world. So a little more about kind of the structure of these compounds. They're extremely persistent, but interestingly, they're also aquatically mobile. So this is just an example of one of these compounds. We'll get into the complexity of the structures here. This is one of the most well-studied. This is PFAS. And kind of a few of the general themes with these structures. They have strong CF bonds, some of the strongest known in nature, so they're really not degradable on kind of geological timescales, as far as we know, haven't been really measured to be degrading by environmental processes. They have this bulky hydrophobic fluorinated chain, and then they also have this polar head group. So generally, a lot of these compounds are strong acids. They're going to be anions at a natural pH in your body or in the water. And they're also highly soluble. So again, they're going to be pretty mobile in aquatic systems. So you might guess from this that there's, myriad sources of these things for human exposure, including, industry, manufacturing of all these products, the actual use of these products, as well as, things like the AFFF, aqueous film forming foams, that are going to go out into the environment, leach from, either landfills or waste or other emission sources. And those can also be transferred to humans. So, there's drinking water, there's diet, there's just environmental exposure, there's indoor dust, there's all different ways that we're exposed to these compounds. So the next question then is, is that bad? What are the effects of these compounds? And there's been a lot of research done on health effects of PFASs. This is a summary from a recent really excellent review by Sue Fenton et al. Kind of summarizing, both the effects that we have pretty well constrained, have pretty good certainty on at this point, which are the bold ones, and then also kind of lower certainty suggested effects. So there's a number of really serious effects of PFASs that have been fairly well established, including thyroid disease, increased cholesterol, liver damage, kidney cancer, testicular cancer, as well as things like reduced response to vaccines, which are highly of concern, especially in the situation we find ourselves in right now. So many adverse health effects. Important thing to note here is that nearly all the studies that are being reviewed in this figure have focused only on the C8 compounds, PFOA and PFAS, which are two of many different PFASs. So there haven't been a ton of studies really delving into the complexity of all of the other PFASs that exist in our environment. Though some early mixture studies have shown increased toxicity in complex mixtures of PFASs. So there's still a lot of work to be done on effects. So getting into kind of, I mentioned PFOA, PFAS, there's so many others. So we're going to look a little bit at how the structure of these compounds can vary. One significant way that they can vary is by chain length. So we talked about the C8s. There's all different chain lengths of PFASs. So you can have shorter chain lengths like a C4 or longer like a C10. And this is referring to the number of perfluorinated carbons in this chain here. There's lots of properties that are impacted by this particular kind of structural characteristic, including hydrophobicity. So longer chain compounds are going to be more hydrophobic. They're going to be less soluble. So your short chains are going to be more aquatically mobile, more aquatically soluble. And also the bioaccumulation factor. So the amount to which this is going to accumulate in your body is chain length dependent. And that's in kind of a more interesting way in that it kind of peaks around the C8s, C6s. It is lower for both short chains and long chains. This is due to the fact that these compounds interact with transporter proteins and proteins in your body. So it's kind of a size-dependent issue there. So we talked about chain length, but there's a lot of other structural variability for these compounds. There's thousands of PFASs, though the toxicity studies, mostly focus on PFOA and PFAS. So the ones that I've been kind of talking about are these perfluoroalkyl sulfonates, which are sitting right here in this figure, summarizing all the different structures. We also have other perfluorinated carboxylates, other compounds that are perfluorinated, as well as polyfluorinated compounds. So there's this whole universe of these compounds. And every time another paper like this reviewing all the PFASs in the environment is published, the number is going up. So here they had said over 3,000. My last check, the number was more like over 8,000 compounds. Another important note when we're talking about PFASs, you'll see a lot of media saying that these are forever chemicals, they never degrade. And I mentioned the CF bond, right, which doesn't break down easily in the environment. They are very persistent because of that perfluorinated moiety. But there are also a number of compounds that are PFASs that are called precursors. And really, if you look at, this giant family tree, a lot of these compounds and a lot of what's produced are precursors. They're not these perfluorinated end products. So PFAS precursors are labile compounds, and they can be transformed into perfluorinated end products via either environmental or biological processes. And the transformation kind of depends on the precursors. Some transform very readily in the environment. Others are fairly stable, but can be metabolized in the body. So this is one of the early studies showing some proof of metabolism of some of the higher precursors to form Pfas, which is a sulfonamide, which is itself also a precursor. So kind of just to summarize this in a simple way, we have things like highly labeled precursors, and then we also have these forever chemicals, right? And the way I usually think about this, so it is an oversimplification, is to think, the really highly substituted precursors that have a lot of alkyl functional groups on them. These are going to get chewed up in the environment a lot more easily for the most part than something that's perfluorinated like a sulfonamide, or one of these end products like the perfluoroalkyl acids, which really don't break down in the environment. And it really is an oversimplification. It depends on what kind of breakdown you're talking about. Some of these highly substituted precursors are still found widespread in surface waters, so they're fairly persistent. It really depends on what kind of processes they're being exposed to, what environment they're exposed to. It's also important to note that metabolic capability varies among species. So there are some aquatic species that can't metabolize these things, whereas mammals can. So you can't really draw broad strokes on how persistent something is, but it's really important to remember that some of these compounds can transform and be metabolized. So now let's move into thinking about what this means for our own exposure and PFASs in our bodies. And this is a really interesting story, I think, kind of starting with the uncovering of PFAS exposure. You can kind of see how it advances with the advancement of analytical chemistry. So in 1968, there was a letter in Nature about a guy, Taves, who did what a lot of scientists did back then. He needed some blood for a study, so he just took his own and wrote a paper on it. Things were different back then. So what he found was he found evidence that there are two forms of fluoride in human serum: inorganic fluorine and also some kind of fluorocarbon molecule. And he did this using ashing. So he basically digested a sample with a combustion type of process and found that the amount of fluoride increased. So it seemed like something was being destroyed, producing more fluoride than he started with. So from there were a number of other papers by Taves and his group throughout kind of the 60s and 70s, where they started to characterize these compounds. So they found that there was some sort of organic fluorocompound of intermediate polarity, tightly bound to plasma albumin in the blood. And they're characterizing this via NMR as a perfluorofatty acid derivative. They found C6 to C8s. And their findings suggested that there were widespread contamination in human tissues, though they had a pretty small sample size. I think they found these compounds in 102 out of 104 people, something like that. But very interesting kind of early work hinting at, the problems that we were going to have in the future. Another thing to note when looking at this is that if you look back at the timelines on what we know now, at the point when they were discovering this in human blood, 3M already had toxicity studies on PFASs. So they already knew that they were getting into people, that they were toxic, and that they were producing them. Not the best scenario there as far as communication goes. So nowadays, with increased, analytical technologies, we know that PFASs are present in the blood of more than 98% of human beings, according to CDC's kind of last surveys as of 2015 and Haynes. And this is a study showing kind of just widespread PFAS concentrations in human blood from general population blood donors from a number of different towns. And what's interesting to note here is you see highest concentrations of PFAS in blood in 2000, and they're going down over time. And what this suggests to me, based on the last slide, is that we are catching after the crest of the wave. So we don't really know what the maximum concentrations here were, but we know they're decreasing now. We didn't really have the analytical capabilities or the knowledge to know that these things were in our blood for quite some time. We also now know some things about the mechanisms of how PFASs bioaccumulate. So they're bioaccumulative in blood, and they also associate with membrane phospholipids. Here's a figure of PFOA interacting with serum albumin. There's a number of different binding sites where it can bind with different affinities. And this, again, was hinted at in those early studies where they found that it was highly associated with serum proteins. So moving forward into kind of the frontiers of what we know, what we're interested in now is, there are studies showing PFAS, PFOA in blood, widespread. What about these other compounds? We saw thousands, on that list. So there's early evidence, a really good paper in 2015 from Vrotander et al., showing that among highly exposed populations like firefighters who are going to have a lot of occupational exposure to these compounds, we see additional PFAS in their blood serum, other than the ones that are kind of regularly in biomonitoring studies. So here you're seeing an extracted ion chromatogram. So they're selecting for specific masses of interest for a exemplary firefighter serum sample. And you've got your high concentration compounds, your C6 and your C8 sulfonates, and then they're kind of magnifying in right on the baseline here to look at the lower concentration compounds that are also present in these blood samples. So they found some shorter chain compounds like a C5 sulfonate, as well as all these interesting substituted sulfonates, chlorinated, ether, ketone, more chlorinated. So this suggests there's more to the story, right? Their study showed that, as far as the relative abundance here, it's pretty low, right? But it's there, and it suggests that there's additional things here that either could be having a biological effect or could act as a biomarker for specific sources of PFAS exposure. So these blue markers here are a control, they're office workers, and they're comparing them to the firefighters, and you can see some of these compounds like this ketone and this chloride are significantly elevated in the firefighters. So it's suggesting, this is a different type of exposure that we're seeing that might, give us some hints as to differences in exposure in these different populations. There's also a lot of studies measuring total organic fluorine, which gives us a lot of really useful information about unmeasured PFASs without having to go and identify each individual one. So this is a study from Young and Mayberry in 2016, looking at the composition of total organic fluorine, extractable organic fluorine really, in blood from residents of 6 Chinese cities. So what they're doing here is they're measuring kind of just the bulk organic fluorine in these blood samples. And then they're also quantifying whichever compounds they can target, like the sulfonates, the carboxylates that we've looked at in previous studies. And these black bars represent everything that they could not explain with their targeted analysis. So that suggests that there's unknown organic fluorine in these samples. Looks like there's some geographic variability where some of these townships had higher contributions than others. And if we look at temporal trends, so this is a more recent paper that came out where they were studying a Swedish cohort that was impacted by AFFF impacted drinking water. And here we see for our bulk organic fluorine, extractable organic fluorine, the percent of this bulk quantity that is accounted for by targeted PFAS is going down over time. So that kind of suggests that maybe, as some of these compounds are being phased out and other ones are brought in, we're understanding less of what's accumulating in our bodies. Relatedly, this kind of third panel here is showing the percent total fluorine accounted for by extractable organic fluorine. So this is suggesting that the portion of organic fluorine is increasing. So there's a lot of organic fluorine, but we don't know what it is, basically. So not a great place to be as far as understanding exposure. So now I want to move into kind of, how all of this comes together in my current research. So what I've been working on is developing comprehensive analysis of PFASs in blood serum from PFAS impacted populations. So I'm going to show you some of the studies I've been working on Firstly, from communities with AFFF impacted drinking water, and then I'll show a little bit of data, preliminary data from recent studies with firefighters. So getting into drinking water, this is a map from a paper in 2019, summarizing 2017 data, of kind of the sources of PFASs in drinking water. The circles are the kind of number of people impacted, the affected community size. And what you can see is there's lots of different sources of PFAS to drinking water. One of the major ones, though, is Air Force bases and airports, which suggests those aqueous film-forming foams, AFFFs. So we're going to focus in on this big blue circle right here, which is in El Paso County, Colorado. And this is a site where toxic PFASs were found in drinking water throughout this area. So this is El Paso County. There's an Air Force base and an airport right up here in the north. And then we have all these townships traveling down south. Security is the northernmost, down to Fountain in the south. So PFOA and PFAS were found exceeding US EPA health advisory levels of 70 ppt in the drinking water in this area from 2013 to 2016. They came in and fixed that. The high levels of exposure ended in August 2015, though we're not actually sure when they began because they weren't being regularly monitored before that. So because of this, I joined a collaborative study called the PFAS Aware Study. That's analysis of water and residential exposure in El Paso County. And to date, there have been very few studies that have really measured levels of PFASs in AFFF impacted communities like this. And those that have really focused on limited PFAS lists of target analytes, 10 to 20 PFASs at most. So the objective here was to do comprehensive characterization of both exposure source and human internal dose to better understand sources and how exposure relates to blood concentration. So we collected raw drinking water samples, that's untreated drinking water, from 38 public and private wells, as well as human serum samples from 213 residents in June 2018. And then collected serum again in 2019 to look at how levels were changing over time. So this is an AFFF source, so I just wanted to remind you these are very complex mixtures, transforming mixtures. They contain a number of precursors in the water, so we're going to kind of get into looking at that. And we know that they're very important for drinking water, not just in this community, but throughout the United States, including a lot of places on the eastern seaboard. They leach into the groundwater. These sites connect as long-standing sources of PFASs for decades after use. So the methods used, I kind of just wanted to quickly touch on, considering that we're using, that we're interested in AFFFs, which are complex mixtures. We really have to try and use methods that are going to be both comprehensive, but also selective for specifically PFAS. So this is a figure that just kind of summarizes all the different analytical techniques that you could use to look at PFAS. From the biggest square, which is the kind of most inclusive, but it's also least selective, right? So you're going to get inorganic fluoride as well as organic if you do particle-induced gamma ray emission. You can use things like NMR. You can use these kind of different bulk methods like the extractable organic fluorine assays. But what we're going to zero in is on this LCHRMS kind of square right here. And I show this just to show you, we're doing non-target, we're getting as comprehensive as we can, but this is still a small square inside of, total organic fluorine. We're not catching everything. So there's always a trade-off between selectivity and inclusivity. And every sample preparation or analytical choice that you make is going to narrow your window. So it's really important to be conscious of that. So for this study, for drinking water, as I said, we collected raw drinking water, untreated, in 2018 from 38 locations. And this was done by whole vessel preparation with large volume injection with pretty good limits of quantitation. Human blood serum was prepared by protein precipitation with online solid phase extraction. We used a weak anion exchange column. So right there, we're selecting only for anionic PFASs, and it's going to work a little better for the longer chains than the shorter chains. And all of these samples were analyzed by liquid chromatography coupled with quadruple time of flight mass spectrometry for that high-res mass spec. So let's get into looking at some of the data. I wanted to start by showing kind of just targeted perfluoroalkyl acid concentrations in raw drinking water. So this is interesting to me because you can really clearly see the gradient, right? We know the source zone was up north, and then these communities down south were experiencing this plume, with the southernmost community having the lowest concentrations. And the other thing to point out here is, you know, the EPA health advisory level is 70 ppt, 70 nanograms per liter. New York is now at 10 ppt for PFOA and PFAS. Most of these levels exceed that. So this cohort was exposed to very high concentrations of PFASs. So how is this reflected then in residents? So for our residents here, this is a study that we published in 2019, led by Kelsey Barton at the Colorado School of Public Health. And she was looking just kind of at the targeted data. So here we're looking at median concentrations of perfluorohexane sulfonate in our different water districts. And we see that in human blood, concentrations were highest in the northernmost community, and then we're lower in our southern water districts. They were also higher in private wells, which is a little hard to tease out because the geographical placement was a little bit variable, but it is interesting to note. One takeaway here, and the reason that in this graph she's looking at perfluorohexane sulfonate specifically, is that this cohort had very high exposure to perfluorohexane sulfonate specifically, which is not a compound that's usually the major spotlight in these human exposure studies. It's usually the PFAS and the PFOA, the C8s. This is a C6 compound. So here we're looking at NHANES general population data, concentrations of hexane sulfonate, some other impacted populations in New Hampshire, Alabama, Minnesota. And in this study, you can see much higher average concentrations of perfluorohexane sulfonate than pretty much most other cohorts that have been studied other than 3M workers. So we had highly elevated hexane sulfonate concentrations, 12 times what was seen in the general population studies, which raises health concerns. Concentrations varied among the water districts, so we know there's geographic variability, and most elevated blood levels were close to the source zone. Some of the determinants, water district of residence, of course, was a determinant for serum PFAS concentrations, as well as frequency of bottled water use, which makes sense. The more bottled water you're drinking in an impacted area like this, the less exposure you're probably going to get. So now let's move on to look at some of the novel compounds that are kind of more where I focus my analytical activities. So one group of novel compounds we were focused on here is the Pfas precursors, which we mentioned. So I found a number of perfluoroalkyl sulfonamides widespread in the groundwater throughout this area in those raw drinking water samples. These have been found previously both in tap water and in AFFF impacted drinking water. I also found a number of kind of more highly substituted compounds that were more sporadically detected, a little bit harder to learn too much about those. So here's kind of what the concentrations of these precursors looked like, going from north, where the source zone was, down to the southern communities. And the asterisk here is because these are semi-quantitative estimate concentrations. We did not have analytical standards for all these compounds. So they're a little bit more estimated concentrations than the other compounds that we target. So we see a steep north-south gradient here in concentrations of these sulfonamides. The C6 specifically was only detected in this northern community and then disappeared as you travel south. And the only targeted compound we had in this list, the C8 Pfosa, made up very little of our total sulfonamides. So without screening for a wider array of compounds, we would have completely missed this story. This kind of gradient, this chain length dependent gradient where C6 was kind of the steepest, and then the C3s, the short chains, seem to travel further. is likely due to differential transport and absorption. Could also be to some extent due to degradation to end products, because these are precursors. What about in human serum? So when we then look at human serum, here we're looking at each of our townships, so north to south, in year 1 serum, year 2 serum, and then in water. And these little hatched marks are for the precursors, right? So I basically summed up precursors that will form a certain compound, and I made the hatches that are the same color as the end product compound. The main takeaway here is just that precursors are certainly present in our raw drinking water, and we do not see them in serum. A couple fluorotelomeres, maybe. The rest of these sulfonamides, we did not see them in human serum. So ready excretion of these compounds, or it could be due to transformation before they're ingested. So if they were transformed in drinking water treatment, we wouldn't have caught that. Biomonitoring a community like this during exposure rather than after mitigation could clarify kind of some of these questions. A little bit more support for some of these kind of possible explanations comes from mouse studies. So this is previous work I did on AFFF dosed mice, where we also saw what looked like rapid precursor excretion or transformation. So in AFFF spiked serum that goes through no biological processes, I saw all sorts of highly substituted precursors, C6s, C5s, C4s, C3s. And then when we looked in the actual blood from mice that ate this same mixture, we don't see all these compounds, right? We just see kind of a little bit of sulfonoamidoacetic acid, C6. We do see some of them coming out in the urine, particularly those sulfonamides we were just talking about in the last slide. All these highly substituted ones, though, we don't see them really come out, and we don't see them accumulate, which to me would suggest they may be readily metabolized to these kind of lower substituted sulfonamides and then excreted. So that might be excretion that we're seeing in this human cohort. So now I want to kind of give a little story about kind of more novel compounds and how novel compound discovery worked in this study. So I detected a peak in these human serum samples in about 80% of people at MZ, mass to charge ratio of 460.933, which matches this formula here. So this formula, this mass and retention time, match a compound that I had an analytical standard for, which is called PFAS. This is perfluoroethyl cyclohexane sulfonic acid, so it's a cyclic compound. But then if we looked at the MS-MS fragmentation, we didn't get a match. So this is a really cool thing you can do with these quadrupole time of flight mass spectrometers. You can actually break molecules apart inside of this instrument in a predictable way that provides a molecular fingerprint for each of these structures. So this is what this PFAS standard looks like. You've got these kind of 3 peaks, with these 2 being really well defined. And this is what the fragmentation looked like for the peak in our serum. We had this 79, which isn't showing up here at all. And then we have all these really low level masses that we did not see in our standard, even though our standard we were able to shoot at a way higher concentration. So you'd think you'd see them if they were there. So this suggests that this is not the same compound. So we ran a concentrated pooled serum sample and targeted the specific mass for really clean fragmentation. And then basically just solved a puzzle for a really long time, trying to look at all the different structures it could be and how they would fragment and which one is the best explanation. And what I found was that the compound that's the most likely that we're seeing here is basically if you take PFAS, remove 2 fluorines and add a double bond. So this is an unsaturated PFOS, unsaturated PFAS. It could be representative of multiple isomers. There could be different unsaturated positions on this thing. Hard to say if there's branching, anything like that. But it's certainly interesting to find it, and it has not previously been identified in human serum, which raises questions of, whether this is present in other cohorts. A quick note, we did see this previously in our mouse studies as well. So this is looking at kind of total comprehensive analysis of serum from AFFF dosed mice and all the different things that we found in that serum. And the circles here correspond to the abundance of the molecule. This giant green circle right here is actually unsaturated PFAS. So interesting kind of corroborating evidence that PFAS may be AFFF associated and bioaccumulative. So kind of to start summarizing what we found in the study, first I kind of wanted to look at how drinking water relates to blood PFAS. So how are things accumulating, what's being enriched in blood, and what's not compared to what we see in the drinking water. So here we're looking at serum to water ratios. So this is the concentration in human serum over the median water concentration in the township. And I'm comparing them to two other literature studies, one for airport exposed workers in 2020, and another one which is an estimated modeling study in 2020 of steady state ratios. That's these orange and blue dots, just to kind of give us a comparison to what might be expected. So we've got all the different compounds here. We've got PFAS and then a number of sulfonates going down in chain length as we go down the list, and then also a couple of carboxylates. And we're looking at the whole distribution in this population of what the enrichment kind of looked like. The black circles are the median in our population. So main takeaways here, we see chain length dependent bioaccumulation, which would be expected in this kind of chain length range. So our C8s are more bioaccumulative, our C3s and C5s seem to be more readily excreted. Ratios are lower than what's been seen in other studies, these orange and blue circles, which suggests to me that our population was still in the uptake phase. They hadn't reached kind of a steady state with this contamination. We also see that PFAS is accumulating in blood to a similar degree to the C8 PFAS, which suggests that it's maybe similarly bioaccumulative to some of these other problematic PFASs. So we see that residential blood concentrations are spatially variable. So from north to south, we have higher concentrations in the northern water district versus the southern water district, both for kind of the known compounds that we study a lot, and also it's hard to see because it's such a low concentration, but also for unsaturated PFAS, this novel compound. We also found temporal variability. So over time, from year 1 to year 2 in our study, we did find decreases in concentrations. For C6 sulfonate, C7, C8, you can see the distribution is decreasing once this problem has been mitigated. We also see this to some extent for this novel compound PFAS. The only compound we didn't really see this for was actually our C5 PFPES. So this is a C5 sulfonate. Should have a really short residence time in the body. So this is an interesting finding that may point to there being other sources of this compound besides just the AFFF impacted drinking water. So now in the last couple minutes, I wanted to quickly show you a little bit of what we're just starting to work on now, which is looking at kind of similar questions among occupationally exposed firefighters. So firefighters have a lot of potential pathways of PFAS exposure. And our questions here are they exposed to novel PFASs? And can we use high-res mass spectrometry to identify novel PFAS biomarkers that can help us understand the sources of PFAS for firefighters? So firefighters are going to have general population exposure, right? Because they're people like all of us. They're going to drink water, have commercial products, eat things, indoor exposures, all of those. And on top of that, a lot of firefighters have worked in the past with AFFFs. So they might have some historical exposure from AFFFs. Additionally, there's been recent studies. There's a very good one that came out in 2020 in ES&T Letters by Graham Peasley's group that show that turnout gear also contains a number of PFASs, particularly precursors and some of the sulfonates, things like that, which is another source of exposure during their work. And also, firefighters have been known to have a lot of problematic exposures for a long time just because of structural fire exposure. They're in these environments where there's a lot of combustion and volatilization, not just of PFASs, but lots of different chemicals. So that could be an additional source. So we're collaborating with Professor Jeff Burgess at the University of Arizona to work on this study where serum has been collected from structural firefighters, both annually and directly after a fire event. Right now we have 18 paired samples to analyze on our QTOF, and we're comparing baseline to post-fire PFAS composition. And this is something my student David Dukes has been working on along with me to get all these samples extracted. So one of the kind of things we've been working on a lot for this project is just developing comprehensive serum analysis. So how can we have a highly sensitive method that selects for PFAS, but is also very inclusive of all these different PFAS with different physical properties. And any sample preparation is going to limit our analysis, right? So we've been working on adding internal standards to our serum. We acidify the serum to denatured proteins. Then we do a protein crash actually in a cartridge for lipid removal. This is an Agilent Captiva EMR lipid cartridge, which works by hydrophobic interactions and size exclusion. So we're not selecting just for anions by this method. Then we transfer to an autosampler vial and run on our QTOF. So some really preliminary data here just to show you kind of the questions we're starting to tackle with this study. We've measured concentrations of more than 40 target PFASs. The ones shown here are just the ones that were frequently detected in the samples, or at least detected in the samples. Levels of the commonly monitored PFASs were fairly similar to general population NHANES levels, and were lower than the Colorado Springs cohort. We didn't see a lot of differences between the post-fire and the baseline as far as concentration of these compounds. What we did see to some extent is higher percent detection in post-fire samples versus baseline samples, including several kind of short-lived precursors as well as short chain compounds, which suggests that might be a biomarker of very recent exposure. Looking at novel compounds, some things that immediately stick out in this cohort is that we're seeing high frequency of detection, again, of this unsaturated or cyclic compound, both in our baseline and our post-fire. This is our bovine serum blank, which has no peak, which is reassuring. Also seeing ketone substituted PFAS. So these are two of those substituted compounds that were also, highlighted in our mouse studies. UPFAS was also found in our Colorado cohort. So it's pointing towards some of these substituted sulfonates possibly being pretty widespread, at least in highly impacted cohorts. This could be associated with AFFF exposure, especially since it's not changing with their exposure to structural fires. And it might just be a long-term exposure that a lot of firefighters have come in contact with. So in conclusion, what have we learned? So we've been working on furthering our understanding of human exposure, particularly in highly impacted populations. We're seeing that humans are exposed to complex mixtures of PFASs from multiple sources, which really complicates our identification of specific sources. Serum detection, we're seeing persistent perfluoroalkyl acids, as well as novel substituted compounds, and some precursors that are likely metabolically labile. Exposure mixture and the resulting blood concentrations are both geographically and temporally variable, which furthers complicating the story. And, it suggests that there may be novel PFAS biomarkers, but some of them, like ones for AFFF exposure, are going to be present in people who drink AFFF impacted drinking water, as well as in firefighters. So it's still not maybe a super specific biomarker of a specific source. So what's next? We're going to continue working on identifying biomarkers of specific exposure sources. So we know PFAS and PFOA have all these different sources. We want to find things that are more specific so we can improve our determination of relative source contributions. One thing to do there is to expand exposure monitoring beyond PFASs, right? So if we just look for, diverse other types of compounds that might help us understand the story. We're also developing these comprehensive biomonitoring techniques trying to close that mass balance on organic fluorine, total organic fluorine, and trying to explain what those compounds are. Because that might offer us some clues, not just about what might be toxic, but also how these compounds behave in the body and the mechanisms of their biological effects. So a lot of what my lab is currently embarking on is to use source materials like AFFFs and metabolic assays to process complex PFAS mixtures to inform high-resolution mass spectrometry libraries, and then use those libraries with all this fragmentation and fingerprinting data to then analyze samples of, AFFF-dosed animal studies, as well as human biomonitoring studies, to really improve how comprehensive our analysis can be. So with that, I just wanted to acknowledge a few people. I have a lot of collaborators on this work, a lot of excellent toxicologists like Jamie DeWitt and Angela Slitt, as well as Jeff Burgess at University of Arizona, and everyone in Colorado at Colorado School of Public Health and Colorado School of Mines, as well as some really great collaborators at Agilent who have helped me with the technical aspects, and my students here at Stony Brook, David, and also undergraduate student Jennifer Marciano, who have been working on processing these samples. And I'd also like to acknowledge my funding support, both from SBU and also from Agilent. So with that, thank you all so much for listening, and I'm happy to take any questions.
That was a great talk, Carrie. Thank you so much. And I have so many questions, and I'll try to just ask the ones that maybe make the most sense. But so with over 8,000 of these compounds out there, and the data, and it sounds like the detection is outpacing the detection of the different compounds, maybe certainly outpacing our comprehensive understanding of them. But I guess I'm just trying to, with 8,000, and obviously I always think about a New York regulation for just 2 of those compounds. I mean, to what extent can we now hone in and say, okay, well these, and I'm thinking specifically about your Colorado study, and that some samples seemingly are excreted, and then other ones are maybe biotransformed. But I mean, where are we in being able to say, cross some off the list and say, well, these are probably biologically irrelevant because they're just going to pass right through us. And these are the ones that are probably, could be worse than PFOA and PFAS. So I guess that's my general question right now, and I'll let you take it from there.
Yeah, so I think, I think about that a lot. I think about that question a lot because it's really dizzying, right? Like, I think even for people who study these, sometimes the complexity of the number of compounds you can look at is overwhelming. And I mean, to some extent, you can say longer chains are going to stay in your body longer, so you're going to end up with higher concentrations overall. But then there's another argument to make that if you're regularly drinking water that has short chains in it, then that's a chronic exposure. And, even though pharmaceuticals don't accumulate in your body, they still have an effect. So who's to say that a C4 PFAS doesn't? There have also been some in vitro studies. I don't know if there's anything in vivo, but there have definitely been in vitro studies showing that some of these precursors that can transform in your body are also more toxic than the end product. So there is toxicity associated even with short-lived compounds that I think we really need to understand that better before we can start really crossing things off. One of the things I've been trying to do recently to start crossing things off a little bit is to look at, kind of that spectrum of persistence, right? And are there compounds that are super highly substituted that just aren't even really going to make it out of the source zone? Like, do we have to worry about everything in AFFF, or are some of those going to transform before they even hit your drinking water and it's just not going to be relevant? I think that's an easy place to start. But from there, I mean, if it's in your blood, I think we have to do further research on it.
Okay, great. Well, I have more, but I'll go silent for a little while here.
Carrie, I'll break the silence here. Do you have any insights on treatment technologies in drinking water?
I mean, there's a number that are starting to work, right? I tend to focus more on the problems than the solutions, which is maybe not the best. But yeah, I mean, I think a lot of what kind of these next gen treatments are starting to, be helpful. And I think a lot of these destructive technologies particularly are really exciting to me in terms of, we could get to a point where we're not just disposing of this stuff somewhere else, but we're actually destroying it. I know Arjun's doing a lot of work on that. I'm starting to join some of those projects as well from the standpoint of kind of just what we were just talking about. Are we just destroying C8s down to shorter chains that are harder to clean up? Or are we really destroying them? So we want to make sure with those destructive technologies that we're really, mineralizing PFAS and not just making C6s from C8s. But I think that's really where the field is going. And that's really the best solution maybe in the end, because otherwise, you either have to incinerate it or you have to store it somewhere, and it's not going to go anywhere.
Yeah, I think everybody's using GAC.
Yeah.
And it's just going someplace else.
Yeah, the GAC, I think the main problem, especially once you look at the full spectrum of compounds, is you're gonna have really quick breakthrough for some of those short chains. So when we saw that pentane sulfonate wasn't decreasing as fast in people's blood as the other compounds, one of the first things we thought was, is it already breaking through now that they've installed advanced treatment in Colorado Springs? We didn't see the C3s or the C4s, so those would have broken through even faster. So our guess is that's not what's happening there, and it's other commercial products. But it's definitely a concern when you see, the C6s and the C5s also being widespread.
Great talk.
Thank you.
Yeah.
Dr. McDonough, I had a question about something you mentioned. Could you talk a little bit more about what you mentioned, that there was difference in concentrations between the samples that you measured versus the bottled water samples? Could you talk a little bit more, because I know that the bottled water comes under the FDA regulations, right? So could you talk a little bit more about how much the concentrations were different?
So we didn't measure the concentrations in the bottled water. I don't know if that study has come out. I think it has. There are some people who are doing bottled water studies, kind of like larger market basket studies. I almost mentioned that, but I was afraid that I was being too chatty and I was going to go too long. But it's definitely important to remember, in a highly impacted community like that, where they have really high concentrations of PFAS in their water, of course it makes sense to drink bottled water. Most bottled water factories are not going to be right next to an AFFF source zone, though some of them do have PFASs in them. Like they're not necessarily much cleaner than your tap water. So if you're not in a highly impacted community where there are high levels, I would say, drinking bottled water is not really likely to decrease your exposure. And it's also, plastic waste and all of these other things. It's really no better unless you're trying to avoid a specific PFAS source.
Thank you.
I really want to answer another question. I said, Carrie, amazing talk, and
Thank you.
And the center and the university is really fortunate to have you as a researcher tackling this very complex issue. So just back to these compounds, New York now regulates 2 of the 8,000 compounds. Some states, like Hawaii, I think, has restrictions on or regs for maybe up to 20. I think California is like a half a dozen. Yeah. So, suppose that you were asked by somebody at the state level in New York, okay, what would be the next set of compounds we should be thinking about for having regulations, and how would we go? Well, maybe I'll just make it started with end it there. So what's the next? Because there's talk about it, and how would you advise people who are thinking about that on what compounds to consider next?
Yeah, I mean, I think it depends for each state kind of what types of exposure they're tackling. And there are, a number of different types of exposures happening in New York. I mean, I definitely think some of these shorter chains, particularly hexane sulfonate, there's a lot of research being done on it right now. It's highly bioaccumulative. It actually has a higher residence time in the body than PFOS, in a lot of studies at least. So that's one that I really think we should be focused on. I mean, it's a hard question because it's like, are you going to go and do a whole study to regulate each and every one of these? Or, are there other strategies? A lot of people say we should just regulate them all as a class. But then the question is, how are you going to monitor for that? Not everyone can do full comprehensive non-target analysis. So, should we be using total organic fluorine for that? It's not as sensitive. So yeah, it's a complex question. But yeah, I would say hexane sulfonate, some of the other sulfonates like that around here might be a concern.
Okay, thank you.
Carrie, it's Stuart. I'll ask one then. How has the medical literature progressed? I mean, you and your colleagues have been making great strides in the last few years. Has the medical community narrowed in on the mechanism in terms of what actually may be causing some of the human illnesses? Is the suspicion that the more it bioaccumulates, the more risk you have, or maybe the shorter chain compounds, even if they don't accumulate as much, could be more reactive? I know it's a big topic, and but is there anything you could say? Is there anything from the medical literature which might help to narrow down the kind of searches that you want to do, or at least inform them? Thank you.
I mean, I think one of the big challenges there is just that there's still so little known on the mechanisms of action, even just for the C8s, that so much of the toxicological studies that are going on are still just trying to unravel, the pathways for PFAS. And I do wonder a little bit if expanding, the analyte list and looking at some different chain lengths and things like that, comparing toxicity more among structures might help with those hints of what those mechanisms are. But it's also, with toxicological studies, it's so complicated and you need so much repetition that you can't study all these compounds. So I think a lot of it is still kind of up in the air. Last I understood, there's a lot of really good work going on about specific compounds. Particularly, there's a lot of really good work coming out of North Carolina, where they have kind of really unique exposures to industrial PFASes, like the Gen-X and the ethers and those novel compounds. There's a lot of really good work being done on those specific compounds and their effects. And I mean, the story we see a lot of the time is that a lot of those replacement compounds that are supposedly less bioaccumulative still do have serious effects that are comparable to what we see for PFAS. So I think that's an important lesson, but there's so much to do. And then that doesn't even get started on mixture toxicity. Mixture toxicity is unpredictable. There've been very few studies. There's not really any satisfying models for what happens when you don't just drink PFAS, but you drink hexane sulfonate and PFAS and PFBS and everything else. And that's all that's happening in the real world, right? We're never just drinking one. So that's a huge area, and people are making great progress, but it's just such a complex problem.
Thank you for that.
I have another question. Hi, Carrie. Nice presentation. Thank you very much. I wonder, are there any plans or on the horizon, are there any substitutes? I mean, it seems like, it reminds me a little bit of the CFCs. At some point, we realized they destroy the ozone layer. So there was a huge, well, investment in finding alternatives. Is there any chance? It seems like we're now aware that these cause problems. And is there an alternatives molecules on the horizon that would do the same jobs in the products that they produce, these compounds?
Yeah, I mean, there's certainly work going into that. On the kind of AFFF front, there are fluorine-free foams now that have recently come out. And there's also just changes in practice that are really important. You know, like there are some essential uses. AFFFs work really well when you're fighting a fuel fire, which is a really important thing to be able to do really well. But you don't have to train with them. You can train with, you know, foams that don't actually work. And, you know, you don't have to use as much of it for things like, you know, stain resistant carpets. And, you know, there are essential uses and then there are non-essential uses. So a lot of the discussions going on right now in, you know, on the regulatory front, I think have to do with what's essential, where do we need these, and where, you know, are they not so important to use. But I mean, they're very good at what they do. So I don't know that there's any compound that can do the same job as well for some of those essential uses.
Thank you.
So Carrie, it's Stuart again. We have gone over, because it was such a good presentation. There are so many questions. I'm sure some of us have more, but hopefully we'll ask you, get a chance in the future. If there's no other questions, if there are no other questions, I suggest we probably just end the meeting. I have the button here. I think I'm capable of ending it.